Yeah, so. Simulated fresh though, we have something. And we're gonna simulate that, so they want to.
What order is Kelly? Kelly is after Justin. Uh-huh. Okay.
Button here. Don't hit
Oh.
that, but yeah, you can hit that and it'll ping all of us.
Okay.
Okay.
All right. I'm trying to figure out where this talk goes. So Fabian is the green one. So green. Okay. So it goes right here. Okay. So I'm just going to move these guys over.
Those new ones?
Yeah, it's the one talk that we did just here.
All right,
Okay.
Let us know if you need anything.
Thank you.
First, was drawn. No, we have to find some way to, we have a secondary, we have to strike out, like, there's a second. I think it's one of them. There's a second facility. I don't know, like, how they, like, I guess I just think they're part of the. So, yeah, like,
And then, it's seven. So like, forget. And then.
Yeah, it was pretty stark. Yeah.
Second machines, I'm gonna sneak away when I can. Sort of thing. Yeah, kind of.
Anything special?
Yeah. Yeah.
Yeah, so, the talks are more, on the program, and what's gonna, you're gonna have someone who's gonna introduce you for the talk. What you're gonna do is just escape whatever talk is there. All the talks are in order of presentation. So if you just, that's, so you are third, I think. That's you. Yeah. So then when you launch your talk, I'll just do it with mine. So when you launch, your talk, just go to slide slow and launch. make sure to come up to display settings and swap display. Because otherwise you'll have the presenter view up there. And then if you want the mouse pointer, it's just Control L, and then you'll have your mouse. And then this will advance your slides as well.
Does this have a pointer on it?
No. Oh, actually, no, it doesn't have a pointer. There was a pointer that they were charging. They may have it charged by the time the talk starts. But if not, we just have it.
Is there someone in here if there's any, because you said you had to run some roads.
I do have to run somewhere else for a short period of time. So yeah, I mean, there's a help button here. So if we have a major disaster, that's the button to hit for help from AV. But hopefully no one's gonna have any issues. But yes, that is
Because I will need the mouse thingy or the mouse pointer, that's all.
okay, do you want the mouse pointer here? Is that fine with the
Yeah,
Control L?
that's fine.
Okay, all right. All right. Well, thank you so much.
I hope so. You're like, HP.
So these are the directions in between speakers. It's escape the talk, close the talk. Once you close the talk, I'm just gonna do it one more time.
Yeah.
Once you close the talk, you then come up to the talks are all in order here.
Perfect.
Once you launch the talk, you just go to
Yeah.
presenter view
Yep.
or present, then go to display settings and swap presenter, because otherwise the notes will get there. And then if they want the pointer, it's Control L. And then this will also advance the slides if they don't want to use the keys.
Got it.
And then if there's anything that happens, it's a problem, help button.
That's easy. Perfect.
Okay, and they'll come running. And then I'm just leaving these here for people. And yeah, I think that's good. What time is it? We have 8 minutes.
No.
Almost no situations. But
So you will be introduced by one person, and then you'll just get right up after Koji speaks for
Yeah.
Sony. And you'll just escape the program. Your slide deck is going to be right there.
Okay.
You'll open it, and then you'll come to
Yeah.
presenter,
That's easy.
and then swap display. So you want to go and swap presenter view, because otherwise the presenter view's on the big screen. So
Okay.
swap display, and then if you want the pointer on the mouse, it is Control L.
Okay.
And then this can also advance the slide. Okay,
11. Like 11:15.
well, you speak at 11:15. Yeah,
Okay. Gotcha.
all right.
Sounds good.
Awesome, thank you. Hold pretty much every presenter. But basically, you'll just hit escape and then close the program. And then all the speakers talk in order. And then when they launch it and then go to the present view.
Okay.
They need to then go up and swap the display
Okay.
because otherwise the presenter display will be on us.
Yeah.
So they have to swap that. And then once they swap it, if they want the mouse pointer
Yeah.
on the screen, it's just control.
Okay. Okay, great.
And if anything happens, I have to do a talk. I
Okay.
have to go somewhere else. If anything happens, there's a help button right here. So if something bad happens, we just,
Okay, sounds great. Thanks so much. This is a great turnout.
yeah.
Yeah. Excited for the day.
Yeah. And it's now 9 and we have a long program, so I'm gonna get started.
Perfect. Sounds good. Thanks for running me through
Yeah.
the instructions.
Hey, good morning. Can everyone hear me? Can you guys hear me? Okay, there we go. Good morning. Welcome to the Northwest Flow Cytometry meeting. This is our second annual Northwest meeting, and I'm very happy to see so many people here today. I'm Michelle Black. I am the director of the Flow Cytometry Core here at Fred Hutch. And Fred Hutch so graciously offers us the opportunity to run this meeting here, which is wonderful that we get the opportunity to have it here. We have a full day program, a lot of good speakers, and we have a poster session. If you didn't see it, it's in the front past or right before the lobby or the security desk. And then the vendors area is on the other side over here. And the program that you guys can all get from the QR code if you want it in your hand. Hopefully, if you need it, we can put it back up. But I don't know if anyone else wants the QR code now. No? Okay. So we're just going to get started. I am going to introduce our first speaker, and we're going to just get this day going. So thank you. So our first speaker today is Andrew Konecny, and he is currently a graduate student in the program of immunology at the University of Washington and performs his research in Martin Perlich's lab at Fred Hutch Cancer Center. His research interests are determining the activation thresholds of mate cells and exploring immune cell population dynamics within the human tissues. In the field of cytometry, he teaches best practices and fundamentals of flow cytometry, develops educational resources and learning opportunities, and creates new flow cytometry panel methodologies. And with that, Andrew.
Thank you so much. Hi. So yes, I'm Andrew. I want to say thank you to the program organizers for inviting me here to speak today. For today's talk, let's see if this will show up. For today's talk, I'm going to really be focusing on high dimensional panel design and just offering some frameworks on how to think about these panels. And today I'll be talking about deep phenotyping panels and versus broad phenotyping panels and the associated error that will eventually limit those panel sizes. So in our lab, we often use what is called, I'm going to call it deep phenotyping panel for today. And these are panels where we have our favorite cell, and we just try to determine all the different phenotyping aspects of that cell. So we're devoting all of our fluorescent space to this, one cell. Some other panels we use are those that are broad phenotyping. So this is when we attempt to determine, all the cells within a complex culture. And in these types of panels, we often have markers that will be mutually exclusive. Our issue is when we're working with deep phenotyping panels and we're trying to resolve every fluorochrome on the same cell, is we quickly come into the problem of spreading error, which has been highly discussed within the literature. And this is when the presence of fluorescence from one fluorochrome interferes with the resolution of another. With broad phenotyping, we can actually make these panels pretty large by getting around spreading error by mutually exclusive markers. But what will eventually limit these panels is a newly appreciated type of error called unmixing dependent spread. Now, neither of these panels are better than the other. These just ask different questions. In our lab, we use them complementary. Often we learn about, say, a new receptor on T cells, and we do all these activation assays for those T cells and determine their phenotype. And then we may go, okay, we want to add more to that story. What cells within the tissue are expressing that receptor ligand that influence that biology? And so these panels have a role together. For each of these, I will give an example of a panel I use in our lab, and then we're going to focus today really on how we get around these two types of errors. So the first panel I'll be showing is a deep phenotyping panel, and for that, my favorite cell type are mate cells. These are an invariant type of T cell, and I like to do activation assays on these and read out all the different phenotypes for them. I also include in this panel some markers for conventional CD4s and 8s so that I can see how they compare to those cell types. And this is just an outline of the gating strategy and all those phenotyping markers that are in this panel. And some panel design considerations I have is with stimulation assays, I'm going to have high antigen density of some of these markers, in particular CD69. And so I'm going to have to deal with them as they will likely cause the spreading error on certain fluorochromes. I'll try to give just outline of metrics for each of these panels. These are just the fluorochromes I used and their antigen assignments and the normalized spectra on the machine I was using. This panel mostly could be used on a conventional cytometer. The only two that are kind of an issue is reading BV420 and B450 at the same time, in addition to probably APC Fire 810. But what is going to be our issue if we're trying to resolve every fluorochrome on the same cell? Well, that is this property called spreading error, and I'm illustrating it here. It's when the presence of fluorescence at high, usually high fluorescent intensities interferes with the resolution of another. And so for this single stain sample, for cells that are positive for BUV661, I have lower resolution of determining cells that are positive for AF647 than if they were negative to begin with. This has been worked out pretty well by Richard Nugent and Mario Roeder back in 2013. So if you're unfamiliar with this concept, I would highly recommend that publication. Now there's ways to predict this type of error and work it into your panel design. This is the spillover spreading error matrix, also described down here. I'm not going to go over how to generate it, just how to read it. For each row, you have a fluorochrome, and it will have some amount of spreading error into another fluorochrome by column. And so BUV661 has a spreading error of 14. It's pretty high into AF647. And that kind of illustrates the event that we're seeing here, that cells that are positive for BUV661 will have a lower resolution of AF647. This property is asymmetric, so we can look at the converse. How much spreading error does AF647 have into BUV661? It's actually pretty low at 1.44. And so we don't see this when we look at our single stain control of AF647. Cells that are positive for that marker do not have a decreased resolution of BUV661. So there's two ways you can deal with spreading error, and I'll use these two fluorochromes to illustrate the first. And that is by taking advantage of co-expression patterns. And so here are those two single stain controls. Here is the full stain sample. And already this antigen assignment is not the worst. On AF647, I put on TCR alpha 7.2, and this is a high antigen density and bimodal antigen. And so I can actually resolve the majority of my double positive cells up here. However, if we know what I'm using this marker for, we can actually get better resolution of our data. I use TCR alpha 7.2 to delineate mate cells by using CD161. So when I look at CD161 cells that are very bright, those actually are very low for CCR7, so they won't have this issue. When I look at my mate cell gate, I can show that when I'm gating mate cells by CD161 and TCR alpha 7.2, I have pretty good resolution of these cells because the spreading error caused by BUV661 CCR7 is pulled into that double negative population. This is a more complex co-expression pattern design. An easier way to go about it is just to put on mutually exclusive markers, say like CD3 and CD19 on offending fluorochromes. Ooh. Okay, so the second way of dealing with spreading error, I will illustrate using BV786, which I placed on CD69. So this fluorochrome you can see has really low SSM values into any other fluorochrome. However, because CD69 will be at very high antigen density, any amount of spreading error that is able to occur will likely occur. So one that I'm of particular interest in is to this APC Fire 810 with the value of 4.8. And we can look at our single stain data and see that, yes, there's actually spreading error occurring. So this is the single stain of BV786 on CD69 at a titer that will make it as bright as possible. And we can see that I have high spreading error of BV786 into BUV805 and APC Fire 810. So this antigen assignment I placed on BUV805 CD45. This likely could be fine because that will appear as a high antigen density that is bimodal and likely will overcome that spreading error. The one I'm worried about is this APC Fire IL-2. This is a continuous staining antigen. And so any loss of resolution due to spreading error will be a loss of resolution of my biology. So I'd like to fix this one. And what we can do is take advantage of the fact that spreading error is proportional to signal intensity. So you will see it increase at higher fluorescent intensities. So if I'm able to, I can use a lower titer of this antibody as long as I'm still able to resolve all of my positive events and decrease that spreading error. So I do better with my assignment over here, having better resolution of CD45. That's fine. That's not my main intent. What I really wanted was to eliminate as much spreading error as I could into this APC Fire 810 to regain all of my IL-2 expression, and that's what we did. So this is with the titer that I went with to solve this issue. So those are the two ways you can deal with spreading error. So I'll just show up some data. So this is how this panel turned out. We have our, quality control gates to get into cells, single cells, live cells, and then T cells by CD3 and CD45. And then my classical mate cell definition by CD161 and TCR alpha 7.2. I use another proxy definition using CD26 by CD161 for mate cells because when I activate them, they actually internalize the TCRs, and that ruins my ability to find them. And then last, we can gate on gamma deltas or CD8s and CD4s, and then the memory populations of these conventional cells by CCR7 and CD45 RA. This is what the panel looked like unstemmed. This is what it looks like stemmed. You can see the loss of that kind of TCR, so I used this definition instead. And so going forward, I'll show some phenotyping data from mate cells and then the memory populations of our CD4s and CD8s. By row, we have our mate cells, our CD4s, and then our CD8s. And then by columns, I have all the phenotyping markers. This is with it out stem. This is it with it stem, and we get good expression of all these markers. So that's it for deep phenotyping. If you're trying to resolve every fluorochrome on the same cell, you'll very quickly come into this issue called spreading error, and you'll have to mitigate it somehow. Now for broad phenotyping, we'll talk about these panels, which you will likely will be much bigger, have more mutually exclusive markers, and will eventually be limited by unmixing dependent spread. We'll go into more of what I mean by that. So for this example, I'm going to be showing OMIP 102, which was a 50 color panel that we put out last year. The main goal of this panel is to have a tool for human samples where you normally just get one shot to look at it. In particular, our lab likes to look at T cells in the context of what antigen presenting cells are. And so we went more into those phenotyping. Some panel design considerations were we wanted to include kind of as many fluorochromes as we could to denote as many cell types as we could. So we needed a way to pick those fluorochromes. And then we also needed a way to determine where error was arising from modeling error in these high dimensional panels. This is an example of all the gating strategy for this panel. So we went highly into the T cell phenotypes, getting into more of our innate types like mate cells, iNKT cells, and gamma deltas, and then into more conventional cell types like CD4 and 8 and Tregs. We are also in the lymphocyte department able to look at B cells and K cells and ILCs. And then for our myeloid cells and dendritic cells, get into types of monocytes, and then our classical dendritic cells, but also able to denote PDCs and basophils. And then for each of these compartments, we included some phenotyping markers to understand some type of their cell status there. Here are all the fluorochromes used in this panel and their antigen assignments, in addition to the normalized spectra. This is kind of all illustrated in this publication down here, if you'd like to look at that more. So one issue we have with these kind of large panels is we don't have this nice sheet that your core provides you with all the bandpass filters on your machine and the dedicated fluorochromes for those. Instead, the question's not, can you run this new fluorochrome that you have on your machine? It's, can you run that new fluorochrome in context of other fluorochromes in your panel? So we need metrics to help you decide which fluorochromes to use. The first one we have is called cosine similarity. This also goes by similarity index or similarity number. And what this is just a one-to-one comparison of how much overlap two fluorochromes are. And this is done by taking the kind of spectral emission of each of these fluorochromes and treating them as one vector, and then lining up the vector of another fluorochrome and seeing what that angle is. If those fluorochromes are totally non-overlapped, they'll have a vector at 90 degrees. Cosine of 90 degrees is 0, so that's a big fat 0 saying there's no overlap. Conversely, as those fluorochromes are more similar, they'll go in vectors of the same direction, and so that angle will get smaller. If that angle is 0, the cosine of 0 is 1, and so that tells you, oh, 1 means complete overlap. Something to note, this is just one number that tells you a relationship about two fluorochromes. It doesn't tell you how much spreading error one fluorochrome has into the other because that is an asymmetric property, and you need to know two numbers for that. So I like pictures, so how does this look like? Well, here's two spectral signatures, one for BUV 496 and one for AF 647. These are totally non-overlapping fluorochromes, so that vector is at a 90 degrees. Cosine of that is 0, so they have cosine similarity of 0. Now I'm showing BUV 563 and BUV 496. These have some overlap, and so their vectors will go in more similar directions. That'll be a smaller angle, and so in this case, they have a cosine similarity of 0.27. So some overlaps happening. In practice, people may just choose a threshold and say, I'm only going to use fluorochromes in my panel of cosine similarity blank and below. And so you can do that. And that's what we're showing over here in our publication, where we computed the cosine similarity of every fluorochrome used. The highest of which we had was between Real Blue 545 and NovaFluor Blue 555 at 0.95. So you can have high similarity and still resolve these. But you may not want to all the time, as we'll talk about later, because some interesting things will happen to your data. So the other number we have to help us decide what fluorochromes to use in our panel is the condition number. And this is commonly called a complexity index or a complexity number. And what this number tries to say is how sensitive is your unmixed data to propagating error that was within your raw fluorescence. And this is just calculated using the normalized spectra from your panel. So in practice, a well-conditioned matrix will have low spectral overlap, small condition number, no modeling error. So whatever error you had within your raw fluorescence will be about the same amount of error in your unmixed data. Conversely, an ill-conditioned matrix will have excessive spectral overlap, high condition number, and then modeling error will be present. So whatever error was within your raw fluorescence will be amplified within your unmixed data. However, this just tells you that modeling error is occurring. It doesn't actually tell you where it occurs. So that's just one number for your panel. So we need something more specific for panel design purposes. However, in practice, people often calculate the condition number of their panel and compare that to other published panels of similar sizes to see how they did. So we can show that for our 50 color panel, we had a condition number of 40, and this did pretty well as compared to other panels that were published of similar size. If we took out 5 fluorochromes, we could actually have that condition number down and take away some of that modeling error. However, this still doesn't say where is the modeling error occurring. And so I haven't said really what modeling error is, so here is an example of it. I'm showing a single stain control TCR alpha 7.2 on AF647, just unmixed and very low complexity panel. This is unmixed by itself with autofluorescence. And this is how I normally think about this fluorochrome and target. However, when I take the same raw data and unmix it in our 50 color panel, we can see that there's higher variance within our negatives, and this expands and limits our detection of the positives. And so this amplification of error is just due to the unmixing matrix. This is the same data. And so in this publication, we kind of dubbed this phenomenon as unmixing dependent spread. And what's really interesting is this is not a global phenomenon. This is localized to specific fluorochromes. And so to show that, we ran CD4 on every fluorochrome as a single stain control and either unmixed that by itself with autofluorescence, which is in the top pane, or in the context of that 50 color unmixing. And we can see that there's some fluorochromes that have that increase in the negatives, like BB700, RB705, or these other 3 that I've highlighted down here. And so we have to think about these colors differently in our panel context. However, there's other fluorochromes like PE-Cy7 that are untouched at all. This fluorochrome looks the same in my 2 color panel versus the 50 color panel. Oh, hi. Yeah. I think we'll get into that in the latter part of the slides. That's okay. But I will answer that question in the end if that's okay. Yeah. Well, to move on, we provided 2 ways of how you can determine how much unmixing dependent spread each fluorochrome is having. And that is just by taking the RSD of those negatives and comparing it and your low complexity versus the high complexity, and so determining that delta. And so in the blue bars, I'm showing the RSD of the negatives for each fluorochrome in that 2 color unmixing, and in the orange, the 50 color. And so you can see there's some like, I think this is BUV 805 that are really not offended. And then we have others like this APC AF647, Spark NIR 685 that all have high unmixing dependent spread. So this is in a fluorochrome agnostic manner. We can also look at it in a way that it is informed by the fluorochrome's brightness by looking at a change in stain index. And then this can help us actually make decisions with our panel design, because we'll have fluorochromes like PE-Cy7 that look exactly the same versus the 2 panels where we'll have AF647, which will appear much dimmer in the context of our 50 color panel. So in practice, to integrate unmixing dependent spread analysis into your panel design, that's what you can do. So you can calculate how much unmixing dependent spread each fluorochrome is having, redistribute those fluorochromes by brightness, and then make your antigen assignments. So APC and AF647 are no longer these fluorochromes in my panel that I consider as very bright. Instead, they're mid to dim. So I'm not going to put on a dim antigen or continuous staining marker. I'm going to put on something that has high antigen density and is also bimodal, like these TCR the alpha chains. The other way you can use unmixing dependent spread in your analysis is to help you decide what fluorochromes you want to use. Where to do that, you can determine like, oh, is this fluorochrome experiencing too much unmixing dependent spread? Maybe I don't want to use that. Maybe I'll slot in something else. And to help with that, we've gone on to try to learn where unmixing dependent spread comes from so that we can provide metrics that can assist in that before running any samples, because generating this other type of data is computationally difficult and running a lot of different controls. So this is unpublished work that I've got to luckily continue on with Peter Mage and Florian Meyer. So I'd ask that even though it's really old math, you don't reproduce. And to learn we're unmixing dependent spread, we kind of have to go back to like what we're basically doing in cytometry. So back in the heyday, we may have done an experiment with 2 fluorochromes that had no overlap. You just measure those fluorescence and you have your data, you're done. However, when we moved into the world of multicolor flow cytometry, we started using fluorochromes that had some overlap. And so we needed methods to determine what amount of fluorescence came from either fluorochrome. Now, a lot of us would say, oh, you just run compensation and unmixing. But what these processes are some type of regression analysis. And we're scientists, we do regression analysis all the time. We have some type of Y that we're trying to explain. In case this is our raw fluorescence of our full stain sample. We try to explain that by some predictors. And these are the single stain information of each fluorochrome. And we know this model will fit every time because we created it with our experimental conditions. What we're solving for is the coefficients. How much does a predictor play into that Y? And so those predictors are our unmixed or compensated data. And with any regression analysis, something we want to avoid is this idea of multicollinearity. That's when several predictors are highly correlated. What this does, it creates higher variance with determining your coefficients. So if we have high spectral overlaps, we would expect to have higher variance within our unmixed or compensated data. And that sounds a lot like unmixing dependent spread. So there's two places multicollinearity can come from. The first is pretty intuitive, and that's when 2 fluorochromes have excessive overlap. So you may have a panel you run with FITC and APC, and that works great. And then you may have another panel with BB515 and APC, and that works great. And you get a new full spectrum flow cytometer, and you're told you can run these at the same time. And you do. And you can resolve them, but you see this kind of higher variance within your data. So you see some amount of unmixing dependent spread. And you go, oh, man, these dyes are just too similar. I just shouldn't use dyes of that similarity anymore. And so we have a number that describes that one-on-one situation. And so you can just decide, oh, these had a 0.98 cosine similarity. I'll use like 0.95 or 0.9 and below moving forward. And then you may not run into this instance again. The harder one to account for is when we have multicollinearity of several fluorochromes overlapping with 1. So here we may have a panel with APC, FITC, APC, Spark NIR, 685, and these 2 have a cosine similarity of 0.64. On your panel, your data looks great. And you may have another version of this panel where instead you're using APC with AF647. These have a higher cosine similarity, but your data still looks fine. And then you have a third panel where you're using AF647 and Spark NIR 685. Again, this has, kind of similar cosine similarity to these pair over here, and your data looks okay. So you've done the transitive property. You're like, oh, I've used all of these together separate. Now I should just be able to use them all together in one panel. However, when you do that, you start to notice this high variance propagating up in your data, and you see a shoot up in your condition number. And it's easy to see from this illustration that, oh, APC and Spark NIR 685 together can explain a lot of the information AF647 is providing. And so this is this type of multicollinearity that's occurring. This is a great illustration, so we need some numbers to actually help us make decisions. And so the number I'm going to be covering is called the SIF value, or standard error inflation factor. So if we're using regression analysis to do our compensation or unmixing, explaining our full stain example by our single stain information, then we can do something similar to understand how much multicollinearity a single fluorochrome is experiencing by just regressing the information of that single stain by all other information of single stains within our panel. I just popped that up here. So with this regression, the better the fit, the more multicollinearity will be occurring for this fluorochrome. So we can take this number 1 over 1 minus the R squared. So how good does that fit work for that model? And this gives us a nice number that it'll start expanding as a better fit. We'll have a higher number, and so that means more multicollinearity. This number is also called a variation inflation factor and has been worked out within the regression literature many years ago. How this relates to our data, though, is if you take the square root of this number up here, you get what is called the standard error inflation factor. And this has a relationship to our data. So the standard error of the coefficient of a predictor variable, this is the standard error of our unmixed data, will be SIF times larger than that if that predictor had no multicollinearity in it at all. So this means that for a larger SIF, for this fluorochrome, we should expect larger unmixing dependent spread. And that is what we see. We can calculate a CIF for each fluorochrome in this panel and plot that by how much unmixing dependent spread it is occurring and have a nice metric to predict that. So this is our measured data and this is our predicted data. So why should everyone love CIFs? Well, this mathematically explains where unmixing dependent spread comes from. It is fluorochrome specific within your panel specific unmixing, where just cosine similarity is a one-to-one comparison, and the condition number is just one metric denoting how much complexity is going on in your panel. The calculation of CIF only requires the normalized spectra, so it's computationally light, and it's also machine agnostic. So I can take data off of any spectrum viewer and start calculating CIFs and panel design processes for any manufacturer. And then last, the literature for VIFs and CIFs was established between the 60s and 90s. This is not a controversial field. It's just how do we apply it to the science that we're doing? Yeah, so that's it for unmixing dependent spread. So now we'll just show some data from this, how this panel turned out. So in this panel, we're able to look at immune cells by CD45, monocytes and macrophages by CD14, take the negatives and go into B cells by CD20 and CD19, and then take those negatives and get into our T cell gates by CD3 and can gate out gamma deltas by that chain. For our B cell phenotype or subsetting, we can go further by IG chains and CD24, get into our T cell flavors, gating out some invariant cell types, these iNKT cells by the alpha 24, J alpha 18, mate cells by the alpha 7.2 and CD161, and then our conventional T cells by CD4 and 8, and our Tregs by CD25 and 127. Out of the lineage negatives of those, we're able to denote NK cells using CD56, and we also included NKP46 and CD16 for further subsetting. And then last, we get into our dendritic cell populations by first getting out basophils that are CD123 positive, HLA-DR negative, PDCs that end up being CD303 positive. And then out of this double negative gate, we get into our conventional dendritic cells by HLA-DR and CD11C, and then the flavors of those by CD141 and FcRn1. We included markers for phenotyping. I'm showing some up here that were specific for more of the T cell phenotyping, and then some down here that were specific for the myeloid and APC compartment. And kind of all of this data is also listed down here in the supplement. So a summary of today's talk. We talked about deep phenotyping. This is when we want to resolve every fluorochrome on the same cell. Very quickly, we'll come into this problem of spreading error, and we'll have to figure out how to mitigate that. When we're working with broad phenotyping panels, these larger panels, eventually these will have some mutually exclusive populations. We can get around spreading error in that manner, but they will eventually be capped due to unmixing dependent spread. So your access to fluorochromes that experience low multi-collinearity in combination that can produce a low condition number and low CIFs is kind of what you'll be limited here. And then likely, these are just frameworks. Our panels likely exist on somewhere in the spectrum of these two. So this is just an idea of how you can think about your panels. And then last, this work has only been possible through strong collaborative effort. And would love, if you've enjoyed this talk, to check out OMIP 102 and look out for our next publication, which will hopefully be soon, which is listed here. Would like to thank my lab. They are a wonderful group of people to work with who are very kind to let me wax and wane about cytometry. In addition to the Lund lab, who's kind of our sister lab, I'm a student through the UW Immunology. Also, I'd like to thank the organizers today again, especially Michelle Black, who also assisted in getting a lot of this data through the kind of cutting room floor. And then Florian Mayer, who works on all of these stuff with me, who used to be in our lab, who's now at Zurich, and then Peter and Aaron. And with that, I'll take any questions. Can I start with you and say, did I answer your question? Okay. You can go with Peter. Mm-hmm. Mm-hmm.
How much of that is place that you might use it one at 70 and a different
Mm-hmm.
that change? What is actually the driving factor
Of that spread? Yeah, it will change if you have a different fluorochrome with different cross laser excitations. It's taking in consideration these CIF values and unmixing dependent spread to explain it by multicollinearity, is how much is those other fluorochromes sitting in the same fluorescent space, and pretty much like the same unique fluorescent space. So for AF647, it's pretty easy to explain by those two because one kind of sits in the first half of that emission and the other one sits in the second half. So if you had a different analog of PE Psi7 that had a different cross laser excitation, and then you had other fluorochromes that were sitting in that space more than the PE Psi7 space, then it would cause more unmixing dependent spread. This is where diagrams help. Yeah, does that answer your question?
It does. I think we have getting at sort of this issue probably isn't.
It is. So it is not, a dye can't just be unique. It needs to also be clean. Yeah, if that's answering your question, yeah. Okay. Yes.
Yeah, you calculated fits. Does that inform which type of markers you want to put?
So we talk about this in the publication is you can make a cutoff of your CIFs and determine, okay, I'm just gonna be as conservative as possible and anything above 2 we'll pick out and try to put in something else. There are fluorochromes that are really bright that can actually experience high amounts of CIF more than others. So AF647 and APC can experience like a CIF of like 5 to 8 and still be usable as in terms of like a bimodal high antigen density marker. If I did that with a really dim fluorochrome like Spark UV 387, it'll kill it. Yeah. Okay. Any other questions? Okay, thank you.
Okay, we're going to get on to our next talk since we have a full day and lots of speakers. Our next speaker is Morten Rizzo. Did I pronounce that right? Morten is a stem cell and immunobiologist in the Fabio Rossi lab at the University of British Columbia. He is investigating how muscle stem cells, progenitors, immune cells coordinate tissue regeneration in response to injury and fibrotic muscle wasting conditions. During his graduate studies, he created an assay for testing therapeutic candidates, modulating cardiomyocyte hypertrophy in muscular dystrophies. As a postdoc fellow at Ottawa, he developed iPS-derived models of human skeletal muscles and is now focusing on mapping immune cell population dynamics during muscle regeneration following injury. Morten is also a technical application specialist at UBC flow cytometry facility, where he helps research teams with antibody panel design and teaches cytometry courses tailored for science in academia and industry. That you can press and point at your screen. Yeah, can. Right,
Okay.
is it working?
Uh,
Or you can do the mouse, whichever you want to do.
Sure.
Okay,
Figure it
thank
out.
you.
Thanks very much. Talk about imposter syndrome following that talk. Being at a very flow heavy meeting, I have a lot of biology to present. So let's get to it. Thank you for still inviting me for this talk. So I'm really excited to present all of this. And I'm going to give you a bit more about the background of muscle, because I'm assuming that there's less interest in muscle tissue within this room. But I am welcoming all feedback. So come and find me, even if you're too scared to ask a question at the end of my talk. Please do come and talk to me because I'm very, very open to feedback when it comes to a lot of the flow stuff. So just as an introduction, muscle tissue is, there we go. So muscle tissue is how we move, how we breathe. It's really important, but often doesn't get the same sort of attention from the research base. But it consists of contractile myofibers, and next to those myofibers are muscle stem cells that are located between the basal lamina and the muscle fiber sarcolemma. They're also known as satellite cells because of their location adjacent to the muscle fibers. In addition to those structural units and the stem cells that rebuild myofibers, we also have vasculature in order to bring in nutrients. We have endothelial cells and vascular smooth muscle and pericytes that are around those blood vessels. We have connective tissue, we have fascia, we have fibroadipogenic progenitors that give rise to fibroblasts as well as adipocytes in muscle tissue. We have tenocytes that build tendons. And then in addition to that, with most interest to this audience, we have a range of immune cells that are tissue resident, as well as those that infiltrate from circulation in response to injury. Muscle has this remarkable capacity to rebuild itself. So when you injure muscle tissue, as shown here with acute injury with a toxin injection, 3 days after injury, you see this complete destruction of the tissue architecture, whereas 3 weeks after injury, you can see that the tissue rebuilds itself, but it can't do it on its own. So although the muscle stem cells are the ones that rebuild the contractile fibers, they need signaling from a whole range of populations that bring in a signaling cascade. So initially, you have lots of granulocytes infiltrating, followed by a wave of monocytes coming in from circulation. Those differentiate into macrophages, first pro-inflammatory, then polarizing into pro-resolving. And then you trigger the proliferation of muscle stem cells and FAPs that then provide that regeneration environment that rebuilds muscle tissue. So our lab has previously published that when things go awry, such as in muscular dystrophies, and immune cells don't provide the right signaling, you can really compromise tissue regeneration. And so there's again, maybe not as in-depth for this audience, but there's different types of immunity. There's type 1 and type 2. And when you have too much of one or the other, that can lead to various disease conditions or fibrosis. And in the context of muscle biology, fibrosis is really important. So I'm researching IL-33, which is part of the type 2 process, or a type 2 immunity. And IL-33 is an alarmin that is normally sequestered in cell nuclei, but upon injury, it can be rapidly released and then trigger signaling through its receptor ST2. And there's a whole range of immune cells that express the ST2 receptor, including basophils, Tregs, ILC2s, Th2s, mast cells. So there's a lot of potential signaling, downstream signaling that could occur, and different paths that could be activated in response to IL-33. So IL-33 can also be sequestered by a soluble decoy receptor that can mop up the ligand within the tissue environment. So what I have noticed from a previously published data set from our lab was when I joined Fabio's team was that IL-33 expression is high in FAPs, the fibroadipogenic progenitors in muscle tissue, pretty stably throughout the injury and regeneration time course. So this is an uninjured muscle and then 1, 2, 3, 4 days after injury. However, FAPs also express the the receptor of IL-33, ST2, and the gene for that is IL-1, RL-1. It's upregulated temporarily at a spike 24 hours after injury, and then is rapidly down regulated. So this was confirmed with single cell sequencing 2, where we can see that at day 1, after acute injury in muscle tissue, you have this upregulation of ST2. So why would a cell express the ligand, but also be susceptible to downstream signaling from its own autocrine expression of the ligand? Actually, it is the soluble decoy receptor that's being expressed in FAPs. So therefore, there is a system whereby the cells can release IL-33 in the context of tissue injury, but they can also express the soluble decoy receptor to dampen the signaling pathway and mop up IL-33 if required within 24 hours after injury. So you can see here that it's the shorter isoform that's being expressed, truncated at the end of these two exons. So in the context of muscle, why is the IL-33 ST2 axis important? It has been shown to play a role in Treg biology. It has also been shown to play a role in ILC2 signaling and triggering eosinophil activation within the muscle fibrosis context. And then also there has been a paper published about macrophage polarization being affected by the ST2 activation. So I wanted to find out what the role of FAP-derived ST2, the soluble form, was in muscle regeneration. And to do that, I employed a conditional knockout system whereby I expressed the Cre recombinase conditionally with tamoxifen injections in a mouse model where Cre was expressed under the PDGFR alpha gene locus, and that is uniquely expressed in FAPs in muscle tissue. And then using LOXP sites in the IL-1, RL-1 gene, which expresses ST2, in order to stop expression of ST2 uniquely in FAPs in muscle tissue. So when I injured muscle in these conditional knockout mice, I found that tissue regeneration at the end of the regeneration cycle was compromised in the conditional knockout mice. As you can see in the curve that is showing smaller muscle fibers in the conditional knockout mice in comparison to the control animals. In addition to that, when doing some first flow, I showed that myogenic cells are found in lower numbers in the tissue in the conditional knockout animals, while FAPs remain fairly constant. And then I started exploring all these different immune populations. So first I looked at macrophage polarization, looking at the pro-inflammatory versus pro-resolving macrophages, and didn't really see much of a difference in the proportions of those populations. I also ran an ELISA to look at eotaxin to see whether there's an attraction for a difference in attraction of eosinophils in the tissue. Didn't see that. I looked at eosinophil activation by flow and didn't really see that. But then subsequently, working my way through all of these immune populations, I ended up thinking that maybe the innate lymphoid cells, which seem to be a powerhouse of triggering really strong immune responses, could be one of the potential players, and they have been shown to play a role in muscular dystrophies already. So I used the IL-17RB GFP reporter mice in order to look for ILC2s in muscle and to optimize a panel without needing to use the reporter. It's just a condition of the system. It has a Cre built into the same construct, so it would have been impossible for me to do the knockout system and have the GFP reporter in the same animals. So I had to optimize a panel independent of the GFP. And then I looked at the conditional knockout mice to see whether we had a difference in ILC2s in response to injury. And I saw that in female mice at 3 days after injury, the ILC2 population was elevated in comparison to the heterozygous controls. And so Why this makes sense is that if you have IL-33 available, you would be attracting ILC2s to be in muscle tissue at higher numbers. If you have the soluble ST2 decoy receptor being expressed from FAPs, then that would dampen that signal, and therefore you would have less ILC2s coming into the tissue. However, in the context of the conditional knockout, we now don't have the soluble decoy receptor. We have stronger IL-33 or more IL-33 present. And in response to that, we now have more ILC2s. So that all makes sense, and that's nice. But the interesting thing that I found was that ILC2s in muscle tissue don't actually proliferate much within the 36 to 72 hour window after injury. So only about 10 to 15 to 20 maximum % of my ILC2 population was EDU positive when I did a proliferation assay. So where are these cells actually coming from is a question that I have. And then another thing, I started using the DMD mouse model in order to look for ILC2 dynamics in muscular dystrophy to kind of follow up on that, because in muscular dystrophy, you have fibrosis that occurs, and I use the DMD mouse model. So DMD is Duchenne muscular dystrophy. It's an X-linked disorder. It's caused by mutations in the DMD gene. That's a very large protein. It is important structurally in order to protect muscle from shear force damage, linking the intracellular actin cytoskeleton to the extracellular matrix through the dystrophin glycoprotein complex. So there's lots of different proteins in this protein complex. So what happens when dystrophin expression is lost, in the case of Duchenne muscular dystrophy, you now lose this connection between the inside and the outside of the muscle fibers. And so your muscle fibers are much more prone to damage. And as a result of that, you have lots of tissue fibrosis and gradual muscle wasting. Patients are usually in wheelchairs by their teens or 20s these days, and there's no real universal cure. There are some therapies that are out there, mostly targeting cohorts of patients, so with exon skipping therapies, et cetera. But there's no real good universal cure still that is readily available. And so the MDX mouse is the mouse model for DMD, and I was looking at ILC2 dynamics within dystrophic MDX mice. And what I found was that in muscular dystrophy, the ILC2 population fails to increase, or I want to say proliferate, but they don't proliferate, which I've just shown because the proliferation proportion is pretty low. So the increase in ILC2s is much lower in dystrophic animals, shown in this plot here. And in addition to that, the ILC2s proliferate even less. So I'm really curious now, why do we not see proliferation in ILC2s? Where are they coming from? So in order to figure out where they're coming from, the first step was, are they coming from circulation? They're mobilized somewhere else, and then they infiltrate into the tissue. So for this, I did parabiotic mouse pairs. So I paired one wild type mouse with a ubiquitously GFP reporter expressing mouse. And then checked for shared circulation. So here the wild type mouse had 40% GFP circulating cells, immune cells, and then the GFP animal had slightly higher proportion of GFP, but basically circulation was joined between these two animals. And then I injured one tibialis anterior muscle of each of these mice. So kept one leg uninjured, and the other leg, I injured the TA muscle. And then I looked at blood ILC2s and TA ILC2s. So there was hardly any cross migration between the two animals, which implied that this migration is not occurring through circulation, because the injured and uninjured muscles in both animals were mostly the ILC2s were matching the genotype of the animal. So although 50% of circulating ILC2s, or not 50, but 40 or thereof, of ILC2s would be either GFP or wild type, the ILC2s that were in muscle tissue were pretty much uniquely based, still the same as what the genotype of that animal was. And so I was able to infiltrate ILC2s to muscle tissue. There was more of them in muscle, but they weren't coming from circulation. So then I looked at when you mobilize ILC2s by doing IL-33 intranasal stimulation. So another parabiosis experiment where one of these mice was not injected, but just I use a pipette tip literally to get the IL-33 solution into the mouse's nose, and then they inhale it, and that stimulates ILC2s within the lung. That mobilizes them to circulation, and then as a result of that, if ILC2s are now circulating, you should be able to see whether they're able to also migrate to muscle tissue. And with this experiment, I showed that there was actually migration from the GFP animals to the wild type animals, not the other way around, because they only mobilized the GFP animals ILC2s from the lung. So that was only the GFP animal received IL-33 intranasally. So here, now you can see that ILC2s do actually end up in the muscle tissue. And so this is a good sign showing that, yes, we're able to attract ILC2s from circulation, but it also shows that in the context of normal immune environment where you don't have an infection occurring in your lungs, you don't attract ILC2s from circulation, and because they're not really circulating. And then I also ran an experiment where I confirmed that IL-33 induced ILC2 infiltration in muscle is dependent on FAP. So I used a diphtheria toxin DTA model expressed by the same Cree system in order to knock out FAPs in mice, and then doing the injury and then seeing that there was way less ILC2s in the injured muscle in comparison to those animals that were wild type for the DTA. So FAPs are required for ILC2 migration to muscle tissue. Okay, so I've done all these experiments, and that's only a handful of immune populations that I have explored so far. And when it comes to ILC2s, I'm already using like 20 plus antibodies per experiment. So for me, the rationale for going spectral was pretty straightforward, right? I have the potential to save sample. I have the potential to run lots more get lots more information out of my experiments by multiplexing the panels. And reagent costs for ILCs, those of you who work with those cells, running 13 color lineage, I may as well get that information already about all of those populations as opposed to just dumping them into the lineage channel. So for me, no brainer. So I ended up also considering it because sometimes I wasn't really getting good resolution for some of my markers. For instance, ST2, I wasn't seeing a high proportion of my ILC2s by conventional flow expressing ST2. I was always running this FMO and then kind of looking at either a histogram or a N/N and not really seeing that ILC2s were expressing ST2. We'll come back to that. It looks much nicer on spectral. And then in addition to that, I was losing some of my T cell markers. So another motivator for me was that apart from some cytoph data and some transcriptome data sets, there was very little information available about immune population dynamics in response to muscle injury. And so, lots of motivators there in terms of the potential for the research field in muscle. So I started off with the Northern Lights. That's the Cytec instrument. We had it in a 3-laser configuration without the UV and without the yellow. That's actually a pretty good setup if you have endogenous reporters such as TDT, because you don't have the yellow laser and therefore you don't need to be reducing the laser intensity in order to still be able to have TDT on scale. But then we upgraded to an Aurora. And with that, my panel also evolved to match the equipment, but also to, as we were getting more and more ambitious, we wanted to get more and more information about out of our panel. And so thank you very much for introducing all of those concepts for everyone. So now I don't really have to go into much detail here, but the complexity index for the panel that I'm showing today was 12.64. I've heard the rule of thumb that you should have your complexity lower than the number of floors. So I'm within that sort of range. And the primary merging that I did was 5 conventional flow panels, putting all of those into one panel, and then getting all that information about all my muscle stem cells, FAPs, subtypes of them, activation profiles of them, plus all of these different immune populations. So if you have any feedback on the sort of markers that I've chosen as well, now that it's been up here for a little while, please do come and talk to me about it. What I am looking forward to, given that this is a sort of flow-focused meeting for the vendors, I'm looking for new floors in specific ranges. I'm sure all of you are. So please take notes. There are some gaps in our abilities to harness the technology to its maximum potential. And then, something brighter in this range would be nice, and a floor that doesn't break the panel in this range as well when you get more complex. So that would be really nice. I know some of the companies, what's coming up next, and I trial new floors as they come out, but I know that these are still areas where we're lacking in development. Okay, so what I really want to show quickly is that flow doesn't always look pretty. And a lot of work goes into those super nice published plots that we see. And if you're a grad student, don't give up, because you can actually make it work. For me, these are just some of the first plots that I generated using a spectral cytometry. It looks nasty, but it gave me hope, because, for instance, I was able to see FAP activation profiles using VCAM that on conventional cytometers, we never really managed to visualize so nicely. We can look at subpopulations of FAPs based off of CD26 expression, and then we can look at the proportions of activation for each of those subtypes of FAPs. This is something that, to me, as a muscle biologist, was like, this is amazing. And so, although it looks messy elsewhere, and I've had to optimize this panel, I was excited from the get-go, and I was sold on the technology, because it now opened so many new opportunities for us to really save our samples and get a lot more information out. Another thing that's really helped me a lot is the autofluorescence extraction that you can do on the Cytec instruments, and I think on other platforms now as well. So you can determine the autofluorescence of individual populations and extract that. And then doing all of this extraction and spillover correction, looking at N/N plots, spending an hour scrolling through the N/N plots. Yes, it takes a while, but it's worth it. You should do it. Retitrating my antibodies. Some of them were too concentrated. So if you're going to go spectral, you might find that your costs are actually going down, because you can get away with using less antibody. And then some of these concepts have already been covered, so I'm not going to go through that, but the gating looks so much nicer after you put in all of that effort. Another factor for me that was important was optimizing enzyme digests. So there's a paper that came out a few years ago now that showed that the longer you digest your tissue, the more you're going to start seeing activation profiles emerging in your population. So that's particularly important in the stem cell field. Me working on muscle stem cells, I'm really invested in trying to keep my stem cells looking like stem cells as much as I can when I'm running my analyses, because that's most in vivo-like when it comes to running my flow experiments. And also important for sorting and downstream analysis if you need to. So one of the issues I was having was that I wasn't able to see CD4 and CD8 resolve in muscle after enzyme digestion. And our gold standard digestion protocol was an hour. So based on this publication, as you can see, that anything beyond 30 minutes is already starting to really introduce a lot of artifacts in terms of transcriptome data. So your PC1, I think it was usually is the most variance that you get out of your sample differences. And so anything below 30 minutes would be preferable. So I had a really ambitious undergrad. Why is this? Where are the bars? None of the bars are actually showing on this plot. This is interesting. all the data's missing. But we ran all these conditions. Trust me, it's there. Show you on my laptop, I guess. But I had a really good summer student who was working with me last year, and he really ran so many different enzyme conditions, and we ended up choosing one that allowed us to reduce our digestion time to 30 minutes, and that also preserved our CD4, CD8, which is super important because now we were actually able to explore all our T cell populations. And in addition to that, look at the effector memory naive profile to look at the activation profile of FAPs, which I've already emphasized. So in the top, the difference here is the enzyme digest, but here it's the uninjured tissue, and then looking at the injured tissue and how much nicer it actually looks now and how much more we're getting out of our experiments. And then in addition to that, I was able to distinguish the tissue resident macrophages from tissue infiltrating macrophages post-injury. Previously, we always lost that ability to look at TIM4 staining on our tissue resident macrophages after injury, but now I'm able to resolve that. And hey, ILC2s are actually all ST2 positive, so that's amazing. You know, kind of really reassuring you and validating some of the things that you maybe had doubts about. Spectral can now really help you as a powerful technology to resolve a lot of the issues. So just to show, we also have an in-house Python-based pipeline in order to run some analysis of the multi-parameter data that we're now generating with so many different conditions. We can do UMAP plots to look at the population clustering of each of those populations that we're pulling out of muscle tissue. And then you can draw these ridge plots to look at ST2 in this example, and to see you know how it's being expressed in all the different populations. So coming back to the biology, so far I've been able to run the experiment with a spectral panel on just female, well, I've now run it on female and male mice, and I'll show a little bit of that data. But here's to confirm ILC2s per milligram tissue. This was the conventional plot that I showed earlier, not quite significant. Very close to that 1 star. And then looking at the spectral data, it's at least replicating the no star, but I will definitely have to repeat this experiment in order to make sure that this is still true. But also we're now getting all this information about all of these other populations and seeing that there are some others that could end up being also affected by the soluble SD2 and lack thereof. So I also like to be open and honest about my data. One of the curious things that I've discovered, and maybe people who work with ILC2s can help me resolve this, is that when I ran this panel on male mice, I saw the opposite trend. And here's a star, which is very annoying, but that's just how biology often is and how immunology also works with males versus females. And very important for you to always run your experiments in both. So if anybody has any ideas for why this could be or should be going in the opposite direction, I'd love to hear your feedback. But just in summary, to show you that we've used this same panel. Once you generate a panel, you can use it for multiple purposes. And so here's another example of how we've used a downsized panel with mostly the same markers in order to validate something else for a different project. So we were looking at non-canonical DGF beta signaling in FAPs. Using a conditional knockout system for TAC1, and that results in eosinophilia. So there's lots of eosinophils coming into the tissue, and also more ILC2s, and we validated that using the same panel, but not with all of the markers. So there's a lot of potential, and it is rewarding once you get something figured out and how to apply that. So future work for me, I'm going to be assessing immune population dynamics in muscle tissue throughout the regeneration time course. I also want to leave some open channels for spike-ins and customizing. So I am looking into freeing up FITC, PE, and APC so that you could use the Legend Plex from BioLegend and you have some endogenous reporter systems and maybe do some proliferation assays. Some limited success with that. I guess you could tell from my floor wish list that I've had some issues with introducing a floor and then it breaks the panel. I don't have as good resolution anymore, so still working on that, but that's the end goal. I really want to look into ILC2 migration if they're not coming from circulation into muscle in response to injury. I've also shown, not data shown in this presentation, that they're not coming from adjacent adipose tissue. They're not coming from the fascia. So where are they coming from? I've done some hematopoietic stem cell transplantation into NSG mice to look at whether ILC2 progenitors going into bone marrow and then proliferating and migrating to tissues, whether they're coming from bone marrow in some weird way, and they can migrate from the bone marrow to muscle, but I was looking at too late of a time point, so I still need to optimize that. And then looking at what happens in the context of dystrophy and how can we resolve dystrophy and maybe come up with some new therapeutic avenues for those conditions. We've also used this panel in vaccine research now. So we have an ongoing project in the team that looks at LNP migration into different immune populations and those immune populations then migrating to the lymph node. So we're also harnessing the same approach to looking at the muscle populations and which cells actually pick up the LNPs when you're vaccinated with lipid nanoparticles. Okay, I'm going to wrap this up and say thank you to the organizers again for giving me this platform, and I'm happy to take some questions. No muscle biologists in the crowd. Cool. Yeah, definitely. We have models for looking at cardiac tissue as well with LAD, and we've done some TAC. So yeah, we could definitely look into that, and that's a great suggestion. I'll look those papers up and read into that more. Thanks. Thanks very much.
Again?
I'll do your introduction.
Oh, yeah.
Yes, and get your slides up. Not sure where they are.
Oh, we hit escape.
Oh, escape.
So it's this flow sorting presentation here.
Okay.
Oh, you have to, you need to look at,
Yeah, oh, there it is. Close.
Yep.
All right, there it is.
And then, yeah, she said that, and then does the pointer work on the screen? Oh, and then she said,
Switch screens.
I can't remember what she said about.
Oh.
Oh, it's that one.
There we go.
There we go.
Awesome. All righty. Well, our next speaker is Dr. Jonathan Fromm. He's from the Associate Professor from the University of Washington. He's a board certified anatomic pathology, clinical pathology, and hematology by the American Board of Pathology, a member of the American Society of Hematology, ASH, International Clinical Cytometry Society, ICCS, and American Clinical Laboratory Physicians and Scientists, ACLPS. Lots of alphabets. And his talk will be flow cytometric cell sorting for molecular testing and FISH alpha case-based approach. So thank you for Dr. Fromm.
Thank you. So this will be something completely different. I'm a hematopathologist, so I spend my time Most of my time looking at clinical cases for patients, about 70% of the time, and spend about 30% of the time doing some translational research. But I was asked to talk about flow sorting in the clinical lab. And as I think you'll see, I mean, this is basically what we do. I have to go run off after this talk is over and look at a flow sort, a clinical flow sort. But for those of you who do sorting, I suspect this will be pretty simple because our sorts are not particularly complex. We just do a lot of them. And so what I thought I would do is present a couple cases, again, in the clinical realm, how we use flow sorting to answer clinical questions. And if nothing else, it'll give you an appreciation of what we're actually doing clinically in flow sort with flow sorting. And again, we're doing it multiple times a day. There we go. Perfect. So this is the outline of my talk. I'm going to review the principles of flow cytometric cell sorting. I think for most of you, I've given this talk before at clinical flow cytometry meetings, but I suspect most of you actually know this at least as well, if not better than I do. And then we'll talk about the utility of clinical flow cytometric cell sorting with some clinical cases. And it really, again, illustrates what we're doing on a daily basis in the clinical lab using this technique, really just as a purification technique for downstream genetic studies on patient material. And then talk about some practical considerations, again, some things that are pretty straightforward, I suspect, for this group. And then finally, summary at the end. So this is a schematic that I made about 10 years ago for flow cytometric cell sorting, talking about flow cytometric cell sorting. And again, I think most of you'd probably know this, but I'm a hematopathologist, so we're looking at T cells, B cells, myeloid blasts. And of course, we have a single cell suspension. In this case, I'm showing a T cell. We have these commercial antibodies, of course, from commercial vendors like Becton Dickinson. And these antibodies are covalently labeled to fluorochromes, and of course the antibody react with a specific antigen. And so when we run these labeled cells through the flow sorter, the individual cells are interrogated with laser light. They of course fluoresce based on the fluorescent molecules that are attached, and then the fluorescent light is collected in these PMTs, and it gives us information about the different populations we have in these very complex biologic specimens. With flow sorting, at least with the instrument that we're using, it's an old ARIA-2, which is being retired, and BD doesn't service it anymore. That's the reason why it's being retired. But the cells are actually put in these individual drops, and the droplet is then charged. And based on our flow sorting gating strategy, and we'll look at those for 2 clinical cases, the instrument can push or pull the drops into these tubes, and our ARIA can sort for different populations at once. It's a 9-color instrument, and again, it's quite a bit less complicated than the ones that you guys probably use here in the Hutch flow core. So why do we do this? So we're typically done to purify populations for molecular fish studies, and it's of course done when these populations are quite small. We're oftentimes asked we see a very small population for a patient with AML, and the population is 0.01%. And so we, of course, have to purify it if we're going to study it by next generation sequencing, or if we're going to try to attempt to do fish studies on that population. And then in the research setting, of course, the indications are the same. You guys know this better than I, where we want to purify a population for for some genetic study, oftentimes next generation sequencing or clonality, to investigate some question. So here's the first example. I have 2. So this is a patient. The ultimate diagnosis was made, and it was using our just conventional clinical flow cytometry along with these sorting experiments and downstream genetic studies, which allowed us to make the diagnosis. And this was actually done by a colleague of mine about 9 years ago, Sian Chen, and she actually published this work as a case report. But the patient was a 63-year-old man with mid-back and leg pain, and he was found to have 40% atypical mononuclear cells on a peripheral smear, and I'll show you an image of what that looks like, but that's one thing as a hematopathologist we spend a lot of time doing, is looking at slides under microscopes, and that also, in addition to flow, is the primary methods for how we make diagnoses. He had no lymphadenopathy or hepatosplenomegaly, and his complete blood cell count, or CBC, showed that he had about 20,000 cells per microliter. Most of these were these two abnormal populations that we'll see in a second. He was also anemic and severely thrombocytopenic. So also they had done serology, and he was negative for CMV and HIV. And then he had an imaging study, CT scan, which showed enlarged mesenteric retroperitoneal lymph nodes and some splenomegaly. And so based on that, and based on his clinical presentation, there was concern that he had lymphoma. I'm not entirely sure why they didn't biopsy a lymph node, probably just because it's easier to do flow the peripheral blood, which we did, and then they also obtained a bone marrow specimen. So this is what we saw under in the peripheral blood, and we see these two populations, two atypical populations. The first of these, small to intermediate sized lymphocytes with clumpy chromatin. It's maybe a little bit hard to tell whether they're neoplastic or not. If you looked around a lot, you could convince yourselves that this population was neoplastic. And this is a kind of a standard stain that we use, a Reichhemse stain. And then there's a second population, which is distinctly abnormal. Just to jump to the conclusion, this is the morphology that we typically see in Burkitt lymphoma. So you see these intermediate to large cells, very dark blue cytoplasm, and vacuoles in the cytoplasm too. So two populations, one distinctly abnormal, it's distinctly neoplastic, the second at least atypical. So let's give a little background about mantle cell lymphoma, since some of you may not be familiar with it. So mantle cell lymphoma is, reasonably common. B-cell lymphoma, about 3 to 10% of all B-cell lymphomas. The cells are small to intermediate in size lymphoid cells that harbor a balanced 11-14 translocation. And the phenotype that we see, and again, we're doing this on a daily basis in our clinical flow cytometry lab. The cells are positive for CD5, negative for CD10, and it's a B-cell lymphoma, so it'll express other B-cell markers like CD19 and CD20 typically. The cells are typically negative for CD10, although rare cases express CD10. I don't know if I've personally seen this, but so they usually almost invariably are CD10 negative. And importantly, because of this translocation, they overexpress cyclin D1. And so we usually confirm that in tissue sections as a hematopathologist. We're doing flow cytometry, but we're also looking at the same specimen but under the microscope, and we do immunohistochemistry. And in a mantle cell lymphoma, it would be considered required to confirm cyclin D1 overexpression. So it's an aggressive lymphoma with a median survival of 3 to 5 years, although recent treatment advances, survival is actually now longer. Very briefly about Burkitt lymphoma. So this is an aggressive germinal center lymphoma. So germinal center lymphomas typically express germinal center markers, not surprisingly, like CD10 and BCL6. And so it's composed of these intermediate or large cells, just like what I showed you in the peripheral blood. And they typically have basophilic cytoplasm and vacuoles, again, very similar to what I showed. It comes in typically 3 varieties: the endemic form, which is seen in Equatorial Africa, the sporadic form, which is what we probably see most here in the United States, and then the immunodeficiency associated Burkitt lymphoma, which is associated with HIV. And I guess if you were in New York City, you might see more of these cases just because of more HIV infected patients. And so this is typically, Burkitt lymphoma is typically associated with extranodal involvement. So in the form in equatorial Africa, it typically involves the jaw. So as pathologists, we see these kind of classic pictures of these kids with this huge, fast-growing mass in their jaw, the kind of prototypic example of Burkitt lymphoma. Abdominal masses in the sporadic form, and then in the immunodeficiency type, nodal and bone marrow involvement. And there can also be leukemic involvement. And so Burkitt lymphoma again is a B-cell lymphoma, so expresses B-cell markers CD19, CD20, markers of germinal center origin, CD10 and BCL6. BCL6 we're not typically doing by flow, we're typically doing it by immunohistochemistry. At least when I've tried to do BCL6 by flow, the antibody fluorochrome combinations that are available don't work particularly well, or at least they didn't 5 or so years ago when I last tried. And then the cells are negative for CD5 and BCL2, and that'll be relatively important later in the talk. And this lymphoma typically shows rearrangements of mix, so they typically show translocation, 814 translocation, 28, or 822 translocations. Again, important for the discussion. So here's some flow data. So this is what we actually saw in the peripheral blood. And these are here all lymphocytes. You know, the color scheme in our lab is T cells in green. Oftentimes a neoplastic population is in aqua. And here, and so that's the population in aqua is CD5 positive, CD19 positive. Again, that's the immunophenotype we would see in mantle cell lymphoma. And then this red population, which is CD19 positive, CD5 negative. But if you look over here, where we've just identified B cells with expression of CD19, the red population expresses 10 and CD38, again, typical of Burkitt lymphoma. In the lab, we typically use surface kappa or lambda light chains as a surrogate for clonality. And you can see both these populations express kappa light chains. We can't really say if they're clonally related because two clonally unrelated lymphomas could both express kappa light chains. And then if we look, again, we can see the population in red is CD10 positive but BCL2 negative, and the population in aqua is BCL2 positive but CD10 negative. I should have said, I'm sorry, I may have misspoke. The population in red, of course, is CD10 positive. And then very briefly, in the blood, the CD5 positive population was about half the white cells, and the CD10 positive population was about 7% of the white cells. And then in the bone marrow, again, we did flow on bone marrow and blood. We see the same populations, but slightly different proportions, or significantly different proportions. So the CD5 positive population is about 8% in the bone marrow, and the CD10 positive population by flow was about 85% of the cells in the bone marrow. And the morphology that we looked at on the microscope, I don't have an image of that, but the showed what one would expect for Burkitt lymphoma. The cells had dark cytoplasm and vacuoles. And because they were so much more numerous than the CD5 positive population, that's really most of what we noticed on our standard stains. My colleague, Su-Yin Chen, then did immunohistochemistry in the tissue sections and showed that those big, large cells were CD10 positive, BCL6 positive, but lacked expression of CD5 and BCL2 with a very high proliferative rate. All of that is completely typical for what we would see with Burkitt lymphoma. However, the cells also expressed cyclin D1, and that's very unusual for Burkitt lymphoma. And so Su-Yin wanted to investigate that further with flow sorting. So what she did was she took from the bone marrow, or I can't remember if it was bone marrow or peripheral blood that we sorted, but she took from the lymphocytes, just based on foreign side scatter, she took the CD5 positive, CD19 positive population in aqua and sorted that on our ARIA, and then separately sorted the CD10 positive, CD38 positive population, specifically from the B cells, to sort the population that we think that looks like Burkitt lymphoma. So these two populations, one that looks like mantle cell lymphoma, one that looks like Burkitt lymphoma. And we did B-cell PCR to prove that they were clonally related, and they did have the same IgH rearrangement. Again, a very common test that the lab is doing. We do this clinically all the time. And then from the flow sorting experiment, we sent the CD5 positive population to the cytogenetics lab. And again, mantle cell lymphoma will have the 1114 translocation. You wouldn't expect it to have an 814 translocation of Burkitt lymphoma, and indeed it didn't. And so everything about this population, from its immunophenotype to its FISH features, are very typical of mantle cell lymphoma. And then from the CD10 positive population, it was 814 positive, which would be typical of Burkitt lymphoma, as I mentioned, but it also had the 1114 translocation of mantle cell lymphoma, and that's distinctly abnormal, very unusual for Burkitt lymphoma. I think this is the only time that I've seen it. So the cytogenetics suggests this population isn't just a typical Burkitt lymphoma population, but it's a mix of both. And so because of that, Su-Yin hypothesized that the patient first had mantle cell lymphoma and then additionally had that second translocation, the 814 translocation, which gave it the Burkitt-like features of that second population. But interestingly, we could still see both populations in the blood and bone marrow. So again, the pathologic diagnosis was mantle cell lymphoma with the second population with Burkitt-like features. And ultimately, interestingly, despite the kind of complex cytogenetics of the second population, the patient was ultimately got an autologous stem cell transplant and was in remission 6 months after transplantation when he was lost to follow-up. So the acquisition of a MYC rearrangement, which in this case, again, was an 814 translocation, can occur in mantle cell lymphoma rarely. Those patients typically have a bad prognosis because the overexpression of cyclin D1 and the 814 translocation together are synergistic and result in a poor prognosis. The immunophenotype of mantle cell lymphoma with MYC translocations, at least as described in the literature, they typically have the immunophenotype of mantle cell lymphoma and typically are without expression of BCL2 and are without these dimorphic these two populations like we saw. So that's what made her case so unusual. And so she actually ended up publishing this as a case report about 9 years ago now. And so the second case I'm going to present is a case of where we use cell sorting to sort the neoplastic cells of classic Hodgkin lymphoma. And so very briefly for this patient, he was a 65-year-old man with a history of polyarteritis nodosa. So this is an inflammatory disease of the blood vessels where he was treated with immunosuppressive drugs, and he developed low-grade fever, mental status changes, anemia, and thrombocytopenia with adenopathy. They didn't want to biopsy the lymph nodes. This is a common theme for the clinicians. They want to see if they can get a diagnosis using a simpler test like performing a bone marrow. And so that's what they did. And they did that on the outside. They did it. They were confused by what the answer was. So they decided to biopsy him again in the bone marrow and send it to us for flow, because as I'll talk about, we actually developed a flow assay for Hodgkin lymphoma, which most labs don't do, although we do it routinely. And then they also sent us, in addition to the specimen, the bone marrow for flow, they sent us a specimen for morphologic examination. So very briefly, classic Hodgkin lymphoma is an unusual type of B-cell lymphoma. The neoplastic cells are rare. These are the Hodgkin and Reed-Sternberg cells. They're usually less than 1% of the cells, and you see this complex inflammatory infiltrate, which is orchestrated by the cytokines that the Hodgkin cells produce. And so we see under the microscope, the first thing you see is lots of lymphocytes, sometimes eosinophils. The Hodgkin cells produce, I think it's IL-5, and so that causes the influx of eosinophils into the lymph node, increased histiocytes and plasma cells. Most labs, in our lab too, are going to make a diagnosis of Hodgkin lymphoma using immunohistochemistry. So the cells are positive for CD15, CD30, PAX5. PAX5 is a B cell transcription factor. Again, we're doing this by immunohistochemistry typically. And then the cells are negative for CD3, CD20, and CD45. And so it's a B cell, it's unusual, it's a B cell lymphoma, but it typically lacks expression of a lot of the B cell markers. And that's because that's involved in the pathogenesis where the B cell program is downregulated in the neoplastic cells. As I mentioned, we developed a flow assay for classic Hodgkin lymphoma. This was in 2007. Actually, we started in 2004 when I was a fellow, and we've been running it clinically since 2007. So we have more than 15 years of experience using it. But the assay uses these 9 antigens, CD5, CD15, CD20, CD30, CD40, CD45, CD64, CD71, and CD95 to identify the populations. And we'll see what that looks like in a bit. And so this is what we saw under the microscope, just to show, it shows some morphology. This is actually immunohistochemistry, but in the counterstain, you can kind of see the large neoplastic cells of Hodgkin lymphoma. They can be multinucleate or mononuclear, really prominent nucleoli. And this is an immunohistochemical stain for CD30, so you can see the CD30 expression over here in this other area, a lot more of these neoplastic cells. As it turns out by flow, we know this population is about 3.5% of the cells in the bone marrow. But again, it had unusual features. It's unusual to have make a diagnosis of classic Hodgkin lymphoma in a bone marrow. And some of the immunophenotypic features were unusual, and it made the outside hematopathologist nervous, so they sent it to us for further study, both by morphology and flow cytometry. So this is the flow study of the bone marrow. And just to point out, we in our lab, by convention, we look at side scatter on a log scale. And so on this plot of CD45 versus side scatter, small lymphs, monos, grands, eosinophils would fall out over here too. And then the population of interest is in red. And it's actually quite large. It's 3.5%. And again, that's quite unusual for Hodgkin lymphoma. These cells are very large compared to the small lymphs, so they have increased side scatter, lots of cytoplasm, lots of cytoplasmic complexity. Cells are positive for CD30. They may even, these cells have a lot of autofluorescence in the FITC channel, so some of this is not real 64 expression, but this is getting up pretty high, and so these cells probably actually do have a little bit of CD64 expression. We've seen this on a couple cases. It's actually, maybe in running this flow assay for more than 15 years, I think I've seen it twice, this case and one other case, so it's pretty uncommon. And most pathologists would think of classic Hodgkin cells as being negative for CD64. On this plot of CD40 versus CD95 in our 9-color assay, here are the normal B cells and macrophages express CD40. Normal B cells here are the Hodgkin cells. Usually they have actually quite bright expression of CD40, so this is a little low. So another really unusual feature of this case. And then the cells have relatively bright expression of CD95. That's actually quite typical. Another unusual feature is the cells lack expression of CD71. They almost invariably express CD71, and yet here it's either low or negative. And then most cases are going to be positive for CD15. And if there's any CD15 here, there may be a little bit, but it's not particularly prominent. And then the cells are negative for CD5 and CD20. That is typical. So really, because they're usually less than 1%, and they're usually less than 0.1%. To identify these cells, we have to have a special tube, and again, that's what we designed back in 2007. But this population was so large that it could be identified in our standard combinations for looking at B and T cell non-Hodgkin lymphoma. And again, we're doing this all the time in the lab. When I get done with this talk, I'll run back to the lab and look at least five or seven cases with looking at B cell tubes for B cell non-Hodgkin lymphoma. So here's the population in our B cell tube. Lacks expression of CD5 and CD19. Remember that I said that this is, being a B cell lymphoma, it's unusual in that it shows this downregulation of the B cell program. No expression of surface light chains. no expression of CD10 and CD20, and no expression of CD38. These cells way up here are plasma cells that are getting in the gates. And then in the T cell tube, the population, again, it's a B cell lymphoma. One can rarely see expression of T cell antigens, and there's complex reasons why that might be the case. But the cells were negative for most T cell antigens, except they did show expression of CD4. Of course, CD4 isn't T cell specific, but it's also unusual to see CD4 expression on the Hodgkin cells. And again, lack expression of CD3 and CD5. And so this just summarizes the immunophenotype. And since we're running a little bit long, I won't go through the entire immunophenotype, but just to note, again, that it had these unusual features, possible expression of CD64, the CD40 expression, which is lower than a typical case of classic Hodgkin lymphoma, and this dim expression of CD4. Under the microscope, another unusual feature, which I think really freaked out the referring hematopathologist, is the cells lack expression of PAX5. Again, a B cell transcription factor. It's the one part of the B cell program that's typically retained in classic Hodgkin lymphoma. And so 90 to 95% of cases are going to show expression of that. And yet this was absent, and the cells had probable expression of CD43, which is not typically associated with classic Hodgkin lymphoma. And so again, those were really unusual features. And then finally, just the percentage of cells, 3.5%, is quite unusual for classic Hodgkin lymphoma. So based on this, it prompted us to then do cell sorting experiments to purify the populations. And then our thought was that we'd purify the population and do B and T cell clonality studies. Again, Hodgkin lymphoma should show a B cell clone. If it was a T cell lymphoma, which is possible because it lacks expression of PAX5 and has expression of CD43, it might have a T cell clone. So that's actually what we did. And so this just shows the gating strategy that we use. The first gate, again, on our ARIA was identifying the population with increased side scatter and low CD40. We then identified them with expression of CD30, CD40, and CD95. Then we identified them with the lack of expression of CD71. Having this kind of low CD15 allowed us to gate out some of the neutrophils which would express CD15. And then finally, because we could, and we had it in the combination that we were using for sorting, we identified the population as lacking expression of CD5 and CD20. And so then very briefly, it was sent for B and T cell PCR, and we found a B cell clone and no T cell clone. So it actually argued to us that it was classic Hodgkin lymphoma. And so that's the diagnosis that we made. And unfortunately, about 2 weeks later, he, because this was so aggressive in him, because of his age, he died before they could actually initiate chemotherapy for classic Hodgkin lymphoma. And so finally, I want to move on to talk about some practical consideration for flow sorting. Again, some of this is probably somewhat superficial for you guys if you're doing really complicated cell sorting experiments here at the Fred Hutch Flow Core. But this was actually done by a master student in our laboratory, Andrew Thomas. And one thing which is perhaps not surprising, and I'm sure you know, is that the ARIA that we were using has 3 nozzle sizes, 70, 100, and 130 micron nozzles. And we were going to do a bunch of sorts, just like I showed for Hodgkin lymphoma, because we were collaborating at the time with Margaret Shipp in Boston, and we were sorting cases for next generation sequencing. So we were essentially using the same strategy for sorting of Hodgkin cells that we used clinically on that previous case, except we were doing it 50 times to sort for NGS. And so Anju was interested in kind of working out the best way to sort these cells. And this has actually been looked at, or had been looked at originally when flow sorting was developed. But what she did was she looked at three populations, megakaryocytes, which are very large, the L428 cell line that we had in the lab, which is a Hodgkin lymphoma cell line, so pretty big cells, and then small T cells. And perhaps not surprisingly, it doesn't really matter what nozzle size we use for T cells, but it really did matter for Hodgkin cells in the megakaryocytes, that the 70 micron nozzle is too small, you're going to shred them, and that the bigger cells, like Hodgkin cells, we got decent recovery with a 100 micron nozzle. And so that's actually ultimately we did with our cell sorting experiments. We didn't use a 130 micron nozzle because, of course, the sorter was unstable, and frequently, when we tried using it, the experiments frequently failed. Second thing that Anju is actually interested in doing, and this is an important practical consideration in the clinical lab, because again, we're sorting these populations and doing B-cell clonality studies, is we're interested in knowing how many cells would we need to sort to see a B-cell clone, because oftentimes we're limited in the amount of material that we have. And so what she actually did was in a model system, she took L428 cells, added normal peripheral blood cells, and then sorted them. And then she found that with one extraction method, and I don't have the kits that she used, but she used one kit, and she was actually able to sort just 50 to 100 cells to get a B-cell clone. And with the other kit, she had to sort 5,000 cells to get a clone. And so at least for us, that was important information, because again, we're doing these kind of sorts all the time. And so knowing what we need to make these experiments work is actually pretty critical. And then finally, again, we know, and others, of course, know that the instrument is pretty unstable with a larger nozzle size. And then finally, one point to make is that when we usually, if we're sorting for clinical work, all we're doing is collecting the specimen in RPMI and then putting it either on a slide if we're interested in looking at it on the microscope, or actually oftentimes we'll collect it in RPMI and then put drops of RPMI, or the Cyto lab will put the drops of RPMI on a slide for fish studies, fluorescence in situ hybridization. And what we invariably see, and others of course have seen this too, is we lose the vast majority of cells that we sort in our processing. But for our purposes, it doesn't really matter, because bone marrow is going to have hundreds of thousands of the cells of interest typically. And so it's actually oftentimes not all that problematic. Might be problematic with a Hodgkin's case, but most of the time we can get away with it. So just to summarize what we talked about. So flow cytometric cell sorting is a common method that we use in the clinical lab for purifying these populations. Again, we do it multiple times a day. It allows downstream genetic testing, which is usually the reason why we're actually sorting the populations in the first place. In our hands, we always, or almost always, develop a custom combination. We're usually doing normal routine clinical flow on the population and sorting it after the fact when the clinician tells us they're interested in doing a sorting experiment. And so we usually have the clinical sorting information in our hands, and then we use that to help guide the sorting experiment. And then, of course, one needs to consider the technical issues that I presented when designing the sorting experiment. I guess with that, I'll be happy to take any questions. Being decommissioned, yeah.
Yes.
Yeah, it's a great question. So we're getting out of the sorting business, which is ironic that I'm getting a sorting talk. Actually, I'm not. I'm gonna, sorting is moving over here to the Hutch. So Su-Yin Chen is going to lead the sorting group. I don't know if you knew that, yeah. And so I'm gonna help Su-Yin do the sorts, because of course I have experience doing it. And of course she's not around all the time. So I'm her backup. So I'll still continue to have a role in the sorts. It's just, it's going to happen here on the Hutch campus as opposed to in our laboratory in the SCCA building. And they actually want to buy the instrument from us and then get a third-party vendor to service it. Because of course BD won't service it anymore. They stopped servicing it at the beginning of the year. So. Other questions? Great, thanks, guys.
Okay, so we have a break for until 11. And then we're going to have our lightning talks with our vendors. So your talk,
Yep.
this
open it.
one, this yours?
Yep.
Yeah.
Okay.
So you're just going to open.
And then the display change. So we
You
go
do that first.
slideshow, and then display
Display
settings,
swap.
swap, enter.
Yep. And then if you want to use just the mouse, it's
Control
Control
L.
L. It's, I wrote it down here.
Okay, thank you. I was listening the first time, it didn't stick.
You know, sometimes it takes repeating.
I'm Jill.
Hi.
Jim Mansfield.
Oh, hi.
Nice to meet you.
Nice to meet you. So,
Some kind of instructions that I also totally
yes,
missed there.
okay, so your talks, what I've been doing is moving the talks that have been presented down here. So that was,
Is there any chance you could download yet another one that I put there? I added something to it. And if that's too much,
do you have it on a thumb drive?
I could put it on one in a second, yeah. I put it back on the, whatever, the chair.
I'm not logged into
Okay.
that. I would have to, so,
I'll put it on a thumb, yeah, put it on the thumb drive.
okay, so we're gonna be, so yeah, you need to do it like, you have 10 minutes.
Right.
Yeah, and then I can just swap you up because you are right here.
Okay, and we
So-
start at 11, is that right? Like, in 10 minutes.
Yeah,
I'll be back.
okay.
I know. Yeah. The event is collected, and we understand. The percentage is very low when we collect the semi stem cell and event is numerous, the stem cell check automatically. We cannot
Hi, can anyone hear besides the people online? You're muted, like the speaker is muted. I don't know if you can hear me. Sorry. Hey, sorry to interrupt again, but the still muted on the virtual. The meeting is still muted for us. We can't hear anything that tells us. Okay, there we go. We're back. Thank you.
We know that, but help is on the way. So, but okay. So ID enables improved discrimination between overlapping foregrounds, and the ID makes switching panel from conventional makes easier or smooth. And also, I would like to highlight ID has a flexibility for future programs. And additionally, 320 laser further increases panel design flexibility. So here are examples of overlapping foregrounds. As you can see here, many overlapping foregrounds can be separated nicely with our optics design and also spectral emission algorithm. And this is a paper from one of our customers, Institut Pasteur in Paris. They have demonstrated a 42 color panel TBNK utilizing 320 benefit. What they found was autofluorescence signature were observed in 320 laser as well. So integration of autofluorescence information from 320 into the emission matrix that increase the stain index. So as you can see here, top plots are from without 320, and then bottoms are from with 320, and all the plots were well separated with 320 information. So standardization is a feature that allows user to set the instrument to an optimized standard setting, ensuring a stable results over an extended period. That standardization is system optimization is achieved by creating a correlation function through daily QC. This is just a one-click QC, and then output can be maintained for long term. More importantly, mixing can be performed after internal correction is done, accounting for difference in voltages between single stain controls and the multi-stain sample. So here is the data to show our stability utilizing our standardization. And the left-hand side has B's data, stability over 175 days. We run 8 peak Bs and calculated and tracked each peak of MFI. And as you can see here, here is 8 peak data. And over 175 days, each peak has less than 5% deviate. So it's very stable. And also on the right-hand side, here we have a 34 color panel data using a TBNK panel, PMMCs, and the MFI and the percent positive of each population was calculated. And also here is animation. I'm not sure you can catch, but actually the plots looks very similar for these experiments. And this is the customer example utilizing standardization. And this is a paper from Odense University in Denmark. And they